Okay, so I'm going to keep on the same thread of looking at the new high-throughput SELEX data from the lab of UC Taipale. And what we did, we compared this data to PBM data, protein binding macroarrays, and suggest improvements to their algorithm. So I don't think I need to convince this audience of the importance of transcription factors and why we want to learn their binding preferences. And the model that we use in this study is the position weight matrix, the very popular model, which has a very nice representation in the logo format, where each position is illustrated here. We have the height that corresponds to the strictness of the position, and we see the variability of the different nucleotides. And just to continue the introduction, one of the great technological advancements of the previous decade in technologies to measure protein DNA binding are protein binding microarrays from the lab of Marta Bulik from Harvard. In this technology, a set of probes that was designed to cover all possible DNA tamers is put on a microarray. The protein binds to these probe sequences, and the intensity is measured using a fluorescence tag. And then computational guys like me can go to the public database of UniProbe and download experimental files that gives us a list of sequences, around 41,000, together with their binding intensities. Now, the study of PBM have been continuing in the recent years, and several methods have been developed to the task of inferring a binding model. And two studies compared between the binding models, one from our lab and one summarizing results of an international competition. And in both these studies, one of the conclusions was that the models are very accurate in predicting PBM binding. So if we learn from one PBM experiment, infer a model, and we try to predict the binding of another experiment on the same protein, we get very high AUC. We are able to predict the binding quite well. But when we take these models and now try to predict in vivo binding, we see that the performance is much worse. It's around an average AUC of 0.7. And of course, there are many reasons for this decrease in performance, such as noisy measurements in cheap experiments and other factors that are involved in vivo binding. But there was still a question if the models that were learned from PBM binding, were they overfitted to the data? Did they learn some of the artifacts and biases of the technology and were not valid for the in vivo domain? So to answer this question, we needed another high-throughput in vitro technology to compare between the measurements of protein binding macroarrays and that technology. And indeed, such a technology came to the rescue, and this is high-throughput SELEX. The previous speaker already introduced this technology, so I won't go over it again, but here I'm showing the implementation from UC Taipale's lab, where we start from random oligos. We see the enrichment in the different sequence files. So again, computational guys like me go to the database and download the sequence files that corresponds to the different cycles of enrichment, where cycle 0 reflects the prior, the background sequences that were in the pool, and from each cycle we are getting a more specific signal. We are seeing a better enrichment of the binding sites. And the number of cycles can vary between 3, 4, 5, and even more. So first I'm going to describe a study that was already published in Nucleic Acid Research comparing between protein binding macroarrays and high-throughput SELEX. And the reason that this was possible is, as the previous speaker noted, there was a recent study 1.5 years ago published from the lab of UC Taipale containing hundreds of high-throughput SELEX experiments on human proteins. And 162 of these proteins also had the PBM experiments. So for the first time, we could really perform a large-scale comparison between these two technologies and see how valid are the in vitro models that we learn. So as the first task, we took the models as published in the paper from Taipale's lab, and we tried to predict PBM binding. We compared it to the models derived from PBM. And what we see is that we get quite good results. The average AUC over 128 PBM profiles is 0.82. So This is quite good. There is a good agreement between the technologies, and as expected, it's not as good as what we would get from PBM-derived models, and here we give the numbers of different methods that infer a binding model from the data. And the disagreement is limited to specific protein families, namely to the SOX proteins and to the zinc fingers. So in these protein families, the two technologies measure slightly different preferences. When we tested on a larger data sets of PBM experiments in which we didn't have unpaired experiments and we performed it in cross-validation, we saw that the results were higher for the high-throughput SELEX models. And this was because in this data set, the protein families were homodomain and ETS proteins. So we concluded for these proteins, there is a high agreement between technologies. Now going on to the second test. The first test was highly dependent on the models that were derived. And it could be that the algorithm produces models that are not so accurate. So we wanted to perform a comparison which is model independent, does not rely on any algorithm to infer these models. So we calculated the correlation between the top 100 ATMers in the different technologies. So we define a set of top 100 ATMers from one technology, and then we calculate the correlation of their scores by the other technology. And we can define ATMer scores in PBM by their average binding intensity. And we use this score in our previous studies, and we know it's quite accurate and robust. For high-throughput SELEX, we used two kinds of score. The first was the frequency, meaning just counting the number of times an ATMer appears in the data. And the second was the ratio, the frequency in cycle I over the frequency in cycle I minus 1. This makes more sense because this gives us the preference of this ATMer going from one enrichment to the next. And these are the results. So we have the four combination of the way we select the 100 ATMers, and we see that when we select them using PBM, we get the highest average correlation. So at least for this task, compared to the data from Typal's lab, it seems that protein binding backarray give a more accurate ranking of the ATMers. And what we found surprising is that when we compare the ratio and the frequency statistic, the frequency is here in red and the ratio is here in blue, we see that the frequency is much more correlated to PBM binding intensity. And we found it surprising, since in the SELEX-SEC data from Richard Mann's lab, they used ratio statistics and got very accurate estimation of the binding intensity. So we went and compared the SELEX-SEC data to high-throughput SELEX from Typal's lab, and we saw that the number of reads in each experiment in SELEX-SEC was tens of millions, compared to this data, in which it was only hundreds of thousands. So our conclusion is that the ratio statistic is very sensitive and requires high read coverage for each k-mer to get accurate estimations. And the last phenomena that we observed is what happens in the last cycle. We see some decrease in the correlation to PBM binding intensity. And we believe that this is due to the phenomena of overspecification, that in the last cycle we see over-enrichment of the high affinity k-mers on the expense of low affinity k-mers, so we see that we don't get as much variability in the signal of the last cycle. Here are a few examples to view in the logo representation, the difference between the technologies. The top models are from high-throughput SELEX. This is the primary model from PBM and the secondary. So here we see one advantage of the high-throughput SELEX technology to measure longer motifs. Another example, for the SOX4 protein, we see that the high-throughput SELEX model resembles the secondary model of the SOX protein. And here we see that there is little resemblance. Maybe there is some similarity between the primary and the high-throughput SELEX model. So far, I've shown you that there is some disagreement, but mostly agreement between the two technologies, but it still doesn't tell us which is more accurate or which is better. So we needed a third technology to be a judge and to tell us which technology is better. And indeed, we used the data of ChIP-seq, and we took it from ENCODE. We took all ChIP-seq experiments that for the proteins that had both a PBM experiment and a high-throughput SELEX experiment. Unfortunately, this left us with only 15 proteins, but still we could test the results. And what we did, we predict chip-seq binding, and what we see here that across the board, high-throughput SELEX models predict chip-seq binding much better than the PBM-derived models. Sorry. So we conclude that these models are better for predicting in vivo binding. Now, why are these models better? So we looked and we saw that we have longer motifs. We wanted to see if these flanking sequences that were measured by the technology do give the advantage. So we kept only the 8 most informative positions for the high-throughput SELEX models, and we see that the advantage is no longer significant. So we believe that the better performance is due to the ability to measure longer motifs. And through our analysis, we also observed a few biases in the data, specifically the data from Typele's lab. And first, we introduced the bias of sticky k-mers. Here, we divided the 8-mers into C-rich 8-mers, which include at least 7 Cs, palindromes, and all the rest. And we see that as we move through the cycles, we see that the median frequency of the C-rich k-mers is increasing. So this is a phenomenon that goes through the experiments. It's regardless of the protein. So the C-rich k-mers are favored by in all experiments, no matter if the protein likes C-rich motifs or doesn't like them. And we see that this phenomena goes on, and this is something that should be taken into account when inferring a model from this data. And this could be due to maybe PCR biases or sequence biases. We don't know the reason, but it exists in the data. Another phenomenon that we observed was false oligos. These are oligos that are most frequent, but they do not contain the binding site. And here for the example, we see the example of this protein. There is one oligo with a count of 14. It doesn't include the binding site. I highlighted the binding site in yellow here. And this oligo goes from a count of 14 to a count of 480. Compared to this oligo, which contains the binding site, it goes from a count of 9 to 62. So we see that these false oligos have extreme amplification, and this goes into more than 32,000. So they have both an extreme amplification, and they seem to have an homogeneous nucleotide composition. So this is another bias that should be taken into account when inferring a binding model. And we believe that these false oligos should be removed either experimentally or computationally. And this leads me to the next part of the study, which is still unpublished, and I have only a couple of minutes, so this is the model reconstruction. And as Gary mentioned before, the models that were built in the study are quite strict. They are based only on k-mers at 1 Hamming distance from the consensus. And in some cases, the seed that was used to build these models, the consensus sequence, was given manually, was selected manually based on prior information. So we want to avoid any manual intervention in the algorithm and to build a model with more variability based on more k-mers. So here we give a few suggestions on how to overcome some of the biases in the technology. First, we say that we choose a cycle using the KL divergence scores, which measure the distance between distribution in the first cycle and cycle I. We choose the first that is above some threshold, so the cycle is specific enough, but not too specific. We also suggest a way to correct k-mer counts to avoid the sticky k-mers, and we observe that the sticky k-mers is a phenomena that is trend-specific, meaning that we see C-rich k-mer, but the G-rich k-mer has very low count. So we can use this in our advantage. And we build a model similar to the way we build in the RAP algorithm for PBM data by aligning the k-mers and constructing a position weight matrix from this model. Here are some preliminary results. So I'll just concentrate here. So we validate the models, different models learned from high-throughput SELEX data by predicting PBM binding and chip-seq binding. For PBM, we see that when we take the all 8-mers model, meaning taking the 8-mers and their frequency, we get the best performance, while we do as good as the published models. When we look at in vivo binding, predicting chip-seq data, the all 8-mers model performs the worst, and the published models perform the best, still better than our algorithm. So I'll conclude the talk. we did a comparison between these two technologies. We observed that they mostly agree, and their disagreements are limited to specific protein families. We detected systematic biases in the data, such as sticky k-mers and false oligos, should be removed to infer a correct model. We saw that the high-throughput SELEX models are better in predicting in vivo binding, while PBM is better in k-mer ranking, at least compared to the data from Typale's lab. And we developed a new algorithm, and still the supervised algorithm that is giving a seed manually performs better, as expected, but our algorithms perform best in the in vivo binding among the unsupervised algorithms. And the all 8-mers model, which has the highest variability and no restrictions, performs best for the in vitro binding. So I thank my advisor, Professor Ron Shamir, and the different funding agencies, and thanks for your attention.
Hi, I had a question about the biases in the initial pool of the SELEX experiment. So we were involved with Richard Mantz, the SELEX-seq thing.
Yeah.
And if you don't, it turns out that there's like at the level, say 12-mers, that it was the right level of the binding, the length of the binding site for the HOX complex. There were like 100 or even 1,000-fold ratios in representation of the sequences in the initial pool.
Right.
We had to model, with Markov models to get any sensible results. So I was just wondering whether the sticky sequences that you were observing might be just the ones that were overrepresented in the initial pool. And also you were using just the frequency of k-mers in the selected pool to get, as an estimate for the affinity. But if you don't normalize by the around zero biases, that is also kind of a dubious.
Right.
Right. So basically two questions. Maybe these two things are.
So first, for the initial pool, we did observe that the frequencies and the distribution for the different k-mers is not uniform, and there are biases. But surprisingly, we saw A-rich k-mers in these pools, while we see the C-rich k-mers enriched through the cycles. So it's two different biases.
Right, but also our biases were actually strand-specific at the Markov model. That doesn't explain it.
And I agree that it is unfortunate. We were trying to model in some way the data from the initial pool, but the read coverage, that's what we hypothesize, is not good enough to include it in the analysis. And that's why we based our models on the frequencies. But I agree that the ratio makes more sense, and you should include the background to deduce a more accurate model. But for this data, we didn't find a way to use it for the initial pool.
We're happy to share the affinity tables, if you want to compare with those. Thanks.
Any other questions? Okay, in which case we'll move on. Thanks, Yaron.